Would it be possible to clone dinosaurs? Is it possible to bring them back to life? Is it possible to recreate a Jurassic Park? A co-founder of Neuralink, which is also co-founded by Elon Musk, the brain implant startup that some time ago embedded a chip in the brain of a monkey, allowing the primate to play video games, has suggested that we have the technology to build a real Jurassic Park. In a tweet, biomedical engineer Max Hodak wrote, "We could probably build Jurassic Park if we wanted to. It wouldn't be genetically authentic dinosaurs, but super exotic novelty species." In the movie Jurassic Park, dinosaurs are created by splicing genetic material found in fossilized amber with amphibian genes to create a complete genome for each species. With what we know today, could we make dinosaurs? To begin with, there are practical questions about the technology, and then there are general moral and ethical questions that are much broader and more complex. Dinosaurs are not genetically authentic like the ones in the real Jurassic Park movie. Created by splicing different genes to create them is the same as creating any other kind of life from scratch. It is a step beyond making a genetically identical clone of a living species. It is even a step beyond cloning an extinct animal from a sample of genetic material. The idea of humans creating super exotic and novel new life forms goes against many people's values. Based on both religious and philosophical ideas, it would be like playing God. Could there be advantages to creating new life forms? Sure, although it is a huge risk that may also have its consequences. And perhaps the most iconic moment in the movie Jurassic Park, Dr. Malcolm says, "Your scientists were so worried about whether they could or couldn't, they didn't stop to think about whether they should." Think of a situation where, perhaps, we discover that dinosaurs have genetic material that makes them immune to cancer in a way that could work as a treatment for people. In that case, perhaps scientists would patch and clone some animals to use in the same way we use the blood of horseshoe crabs, for example, to help make vaccines. Whether you agree with this idea or not, you can see that someone could argue for it and make a compelling case for cloning the dinosaurs we need. But that's not the end of Jurassic Park, whose goal is surely to create a luxury destination for extremely wealthy people. Will we ever see a real Jurassic World? Not as we see it in the movie, but some paleontologists are exploring another way to revive dinosaur traits, through genetic engineering that produces birds more like their dinosaur ancestors. Genetic engineering is making birds look more and more like dinosaurs. The way the geneticists in Jurassic Park made their dinosaurs, it would never be a reality. The genetic material just doesn't last long enough. DNA begins to decay at death, and, given its rate of disintegration, even under ideal conditions, there is little hope of obtaining even a shred of DNA from fossils older than 6 to 8 million years. It's too recent for Tyrannosaurus and Triceratops, let alone earlier dinosaurs that were born between 66 and 235 million years ago. Fossils can preserve the structure of collagen fibers and blood cells. Instead of bringing ancient DNA back to life, scientists are thinking about how to go backwards from modern DNA. Birds are living dinosaurs, true. They are nothing like a Diplodocus or a Spinosaurus, but they are another line of dinosaurs that evolved about 150 million years ago and survived while all their relatives went extinct. And since bird genes retain traces of their prehistoric past, as does anatomy, some scientists are looking to these living dinosaurs to reconstruct the lost animals that came before them. Paleontologist Jack Horner, a consultant for the Jurassic Park films from the beginning, has had a lot of press to introduce this concept as the chickensaur project. By tweaking genes and developing chickens that Horner and others have proposed, scientists can create a bird that looks like a velociraptor. After discovering that the toe perched on the legs of modern birds begins to develop as soon as embryos begin to move their muscles, biologists paralyzed that toe in developing birds and found that it retained the ancestral anatomy seen in non-avian dinosaurs. Geneticists at Harvard University announced that they could create experimental chickens without beaks and with jaws more like those of dinosaurs such as Velociraptor, with a few more changes, including a long, balancing tail. Scientists will be pretty close to producing what is going to look like a Cretaceous rooster. This method will not allow us to bring back Velociraptor or Tyrannosaurus. Those dinosaurs are behind us and are never coming back. Instead, a chickenosaur would be closer to Indominus Rex, the fictional villain of Jurassic World and a hybrid of various dinosaur genes and other creatures. Or even genetically modified elephants would look like mammoths. It would not be time for resurrection, but it would be time for reinvention. However, the point is not that someone will ever present a live chickenosaur on television. The real goal of these studies is to understand the relationship between genetic change, development, and anatomy that accentuated one of the most wonderful evolutionary transitions of all time, taken together. All of this could reveal what happened to genes and anatomy as birds proceeded from their ancestors. 100% of a dinosaur can be found in the DNA of a chicken. This is because chickens and all birds are dinosaurs. It turns out that they are not only descendants of dinosaurs, they are actually dinosaurs. Most of what people believed about dinosaurs turned out to be wrong. When the amniotes first evolved, they were divided from the beginning into two main groups: the sauropsids and the synapsids. The sauropsids are divided into two further groups, anapsids and diapsids. These names are based on the number of holes in the temples of the skulls. By the way, all anapsids eventually became extinct. The diapsids evolved into several diverse groups: lepidosaurs, lizards, snakes, tuataras, some marine reptiles, testudines, turtles, and archosaurs. The name means ruling reptiles. Crocodiles, pterosaurs, and dinosaurs. Synapsids evolved into several groups as well, but the only one that survived was Mammalia. The first mammals and the first dinosaurs evolved at about the same time. Both were mesothermic or endothermic. Both, therefore, needed insulation. The first protofeathers actually evolved in archosaurs before the split between dinosaurs and pterosaurs. Therefore, both lineages had feathers. Primitive down feathers, not just pycnofibers, have now been found in A. Pterosaur feathers are basal for the Dinosauria group. So it is a warm-blooded being, and being hollow-boned is also something dinosaurs evolved early on. The huge sauropods can attribute part of their enormous size to the strength of their hollow but light bones. The fact that many large dinosaurs were covered with scales is probably just a result of being huge. Large animals in warm environments tend to have sparse body cover. Elephants, rhinos, etc. Tyrannosaurus Rex was, by all evidence, completely scaly. The avian branch of dinosaurs is part of a group of dinosaurs called Maniraptors. This group includes dromaeosaurs, raptors. You can clearly see that birds are not really special at all. In fact, birds are no more different from other dinosaurs than bats are from other mammals. They have adaptations to flight and skeletal changes to support it, but in reality, they have fewer changes than, say, a dolphin from a monkey. If we consider bats, dolphins, and monkeys to be mammals, we should consider birds to be just another dinosaur. If you are trying to build a different species of dinosaur from a modern avian dinosaur, you are pretty much doomed to failure. It would be like trying to engineer a deer using human DNA. You already have a dinosaur. That should be appreciated. They didn't all go extinct. A whole branch of them survived, just as a whole branch of mammals survived. Bones provide the roadmap, but living bird features can give some step-by-step directions to get from one point to another. Can know a lot about dinosaurs because some of them are still with us. Let's look at chickadees, quail, crows, crocodiles. Still live in a Jurassic world. Australian scientists have conducted a study that determines that all the links in the DNA chain, preserved at the ideal temperature of 5 degrees Celsius, would be completely destroyed after 6.8 million years, which is incompatible with the idea of finding intact genetic material in a dinosaur fossil from 80 million years ago. Environmental conditions, such as temperature or soil chemistry, and even the time of year when the animal died, can affect the process of DNA deterioration. Despite all the current technology and scientific advances that exist, DNA samples only remain useful for approximately 1 million years. This means that, theoretically, we could clone a Neanderthal, but not a Tyrannosaurus Rex. After an animal dies, its remains fall to the surface where sediments are constantly being deposited. The soft tissues of the deceased animal degrade or are eaten by scavengers until only the hard tissues of its anatomy remain, and new sediment, mud, dust, sand, pile up until it is buried. Water from rain, or from some water source near the place where the animal died, seeps through the sediments over the years, dissolving minerals such as silica. These sediments, the product of erosion or weathering of pre-existing rocks, is what serves as protection to prevent the bones from disintegrating and turning into rock. Subsequently, when the water evaporates, the minerals precipitate in the hollows of the spongy tissue of the bones until they harden, a phase known as mineralization. This process takes place molecule by molecule over a long period of time, until the body is completely mineralized, turned to stone. The climatic conditions of the place where the animal died change. It is likely that the erosion will expose the fossil to the open air where it will be discovered by paleontologists. Can DNA be recovered from a fossil? No, fossils do not contain DNA. At least not recoverable DNA in an optimal state for the process we are considering. When an organism dies, the complex chains of molecules that form the DNA begin to disintegrate due to enzymes, so that they lose an increasing amount of information as time goes by. As the damaged DNA strands lose all the information needed to produce an embryo after being introduced into an empty egg, it is impossible to clone a long-dead animal using the degraded DNA from its remains. It was calculated that DNA has a half-life of 521 years. This means that half of the chemical bonds in any given DNA sample will have broken 521 years after the death of the organism, and half of what is left will disappear over the next 521 years, and so on until the molecule eventually disappears completely, some 6.8 million years later. What happens to the younger fossils? DNA recovery techniques are very advanced, and it has now been possible to obtain samples from remains thousands of years old because they still contain some recoverable DNA. In fact, in 2013, the complete genome of Neanderthal man was recovered. However, this DNA is young as it is only 38,000 years old. DNA is a stable strand, so it can sometimes be recovered directly from the fossil, as was the case with this and other Neanderthals or several mummies that are only between 3,000 and 40,000 years old. What about mosquitoes in amber that have dinosaur DNA? Although an amusement park full of dinosaurs might be the dream of many people, it is something that will remain a mere fantasy and on the big screen. It seems that the digestive tract of a hematophagous insect is not the best place to find DNA in good conditions. Let's not forget that these insects feed on blood and their enzymes are prepared to break down the main proteins found in blood. DNA is one of them, and the acid pH of a digestive tract would accelerate its deterioration. Besides, although these creatures evolved to drink blood, they were not able to penetrate the tough skin of most dinosaurs. Most likely mosquitoes bit dinosaurs in sensitive areas such as eyelids, wounds, ears. A mosquito will bite not only one individual, but if it can, it will bite several, and when conditions are right it will even feed on several species. The problem here is that for this DNA in amber to work, our mosquito must have bitten only one specimen before being trapped. One of the big problems with studying DNA, and especially ancient DNA, is contamination with DNA from modern bugs. For example, when the Neanderthal genome was sequenced, many scientists doubted that it was actually the DNA of this extinct man, and it was thought that it was contamination with DNA from the personnel who worked and handled the fossil remains. It is clear that we would not confuse human DNA with dinosaur DNA. But let's think about other sources of contamination. The DNA we extracted from amber could be from the mosquito, or from various prey of the mosquito, such as different individuals of the same species or different species. We do not know the sequence of dinosaur DNA. How do we expect to be able to distinguish DNA from one species from another? What's more, the ancient genetic material could also be from the mosquito's gut flora, from its parasites, from the tree itself that generated the resin, or from amber-eating bacteria or other inadvertent microscopic passengers that got trapped. Even if we obtained DNA sequences from a single dinosaur, would that be enough? DNA is by no means small. For those who remember, one strand of DNA contains a total of 3 billion genetic codes. To expect to get one from a single specimen is beyond the scope of modern science. Is one thing to have the sequence and another to have the DNA. Contrary to what one might believe, a sequence is not the DNA itself. It is a digital file that tells us the order of the nucleotides, building blocks of DNA. We would have to manufacture artificial DNA from that information. Can it be done? Yes, in 2007 scientists developed the first artificial genome, and in 2010 they introduced it to an organism without DNA to see if it worked, and they succeeded. Of course, this was a bacterium and does not compare to a dinosaur genome. But in theory it is possible to take a code and recreate it. DNA in an animal exists in an ordered form in discrete structures called chromosomes. What scientists should do next is create individual chromosomes to complete the karyotype, complete set of chromosomes, and thus have the proper instructions for making a baby dinosaur. Suppose we get around all the obstacles so far presented and have a replica of a dinosaur cell nucleus, ready to be inserted into a donor egg and begin its division to form a baby dinosaur. Which egg to use? Dinosaurs have two living relatives, crocodiles and birds. The most logical thing to do would be to use bird eggs, and logically we would not use hummingbird eggs, but from something much larger, perhaps an ostrich. After all, the larger dinosaurs laid eggs larger than the size of a volleyball. Let's hope for the best and that today's larger egg is enough for our baby dinosaur. Other problems could be the thickness and structure of the shell, but the truth is that eggs from many dinosaurs are known and they have a similar structure to avian eggs, so our hatchling would breathe without a problem. But no. The technique used to extract the base strand of DNA in Jurassic Park is not feasible. Let's remember that in this science fiction movie, the DNA of the dinosaurs is obtained from mosquitoes that were trapped by the amber of the trees, thus becoming fossilized. First of all, there is no real case in which someone has found dinosaur blood inside a mosquito trapped in amber. Basically because the DNA contained in all insects that have been found in this way belongs solely to the insect and not those dinosaurs that served as food for these mosquitoes. By definition, two organisms are clones when they are identical, which in this context implies that by necessity they have practically the same genetic information. So the first thing we need to generate a clone is all the genetic material of the organism to be cloned. Here is the main problem when it comes to cloning a dinosaur. It is impossible to get that genetic material, fossil remains of dinosaurs, like the skeletons we see in museums. Our petrified materials where all organic elements have disappeared. The basic cloning process that was used to, in 1996, achieve the first cloned mammal, Dolly the sheep. A tissue sample was taken from the sheep they wanted to clone, the cell nucleus was extracted from it, where almost all the animal's genetic information is found, and an egg was obtained from a second sheep from which the nucleus was removed. The next step, very briefly, consists of introducing the nucleus of the sheep to be cloned inside the empty ovule, and the cell obtained from this union is used to impregnate a third sheep, which gestates the ovule and finally ends up giving light to Dolly. As a curiosity in this story, Dolly's name was chosen in honor of the famous singer Dolly Parton. In the case of dinosaurs, instead of an egg we would use an unfertilized egg, but the procedure would be similar. However, here it is essential to get the genetic material intact and within a nucleus so that we are able to transfer it to the egg. Since if we only have the genetic sequence, the situation is complicated. This is what happens with the theoretical approach of cloning a mammal from which it has been possible to recover organic material with sufficient quality to sequence the genome, but not with sufficient quality to have an original, complete genetic piece within a cell nucleus. Just having the sequence greatly increases the difficulty since, among other things, it means artificially manufacturing the entire genome and, although this is possible on a small scale, if we are talking about animals as complex as a mammal, that would be a practically insurmountable task with the current technology. So today, cloning complex organisms is a task that ranges from being somewhat difficult but technically feasible, to an impossible task depending on the living being we want to clone.